Thank you, Dr. Tang, for
Mhm.
the introduction. So, okay, let me got it. Today, the afternoon section of this workshop, like Dr. Tang said, I'll be talking on the next generation sequencing, and I'll just be introducing some brief concepts. So let me make this a little bit bigger. All right, all right. I know Dr. Tang talked about how the technology driving the sequencing of genome, human, including all other animals, is really moving at a very rapid pace. And as you can see, it's not really parallel to the Moore's law. So the technology is going faster than predicted by the Moore's law. So as you can see, looking at the cost of sequencing a genome has really dropped as the technology becomes more advanced. And the interesting part of this is when the first Human Genome Project was put forward, there was only they used the Sanger sequencing technique, which is a traditional way of sequencing the genome. Of course, it takes a longer time and all that. It took about 13 years to do that, to complete that. So the Sanger sequencing, which is this is just a brief explanation of how this method, how it works. First is to have your DNA fragment, and then with the application of heat, you the nature of the DNA, and then which separates the complementary strand, and then you take the what's it called? You take a primer, attach it to the 5' end of the DNA you're trying to amplify, and then you amplify your DNA, and then with the use of a DDNTP, which is a dideoxy NTPs, you use that which has a fluorescent label tag to them. So that is what they use in actually sequencing or detecting the sequence um, of a DNA. And this process is slow, it takes forever, and then also the chances of error is also high. But with the advent of new technology, for example, the Illumina, we begin to use the next generation sequencing, which uses a different type of application method, which is known as the bridge amplification. And that, unlike what's it called, the Sanger, that uses the fragment and then the tagged DNTP. The next generation sequencing uses what is known as an adaptor ligation, which is attached at the ends of the DNA fragments, and then that is also read through um, an array, or sometimes they put it on for the bridge application to happen. So the NGS over time has actually reduced the time required to sequence a genome. And interestingly, recently this year, a record was being set. I think the human genome of a patient was actually sequenced for 5 hours and 2 minutes. So that is actually in the Guinness Book of Record at the moment, and that was also helpful because after they were able to sequence the genome of that patient, they're able to find a genetic variant, which helped them to actually detect the diagnosis, which is heart related. So they were able to perform a heart transplant before, which saved the patient's life. So we see how important this technology and this concept is to medicine, and also to the research setting as well. So like I likely said, the cost for human genome sequencing is really going down as technology improves. So this is just a flow how the NGS analysis actually works. So you have either the DNA, RNA, or the protein sample, and then you have your sequencer, which could be from any of those company. We have the Oxford Nanopore, we have the Illumina, and all that. So once you have your DNA sequence, and then this is where the bioinformatics aspect comes into place. So you begin to do the assembly, you can do the alignment, and these also are very useful for you to be able to detect variant and also new transcripts and also DNA protein interactions. And this could also help in finding the genetic component of some complex diseases like the mental disorders and cancer. And just like the example I gave, that happened this year, early January. So these are some of the companies, and these are some of the NGS sequence that are being used in the industry. So let me see. So the Illumina sequencing, which is also one of the companies driving the technology regarding DNA sequencing. This is just the flow work of what they do. They prepare the library, which is fragmenting the DNA, and then they ligate the ends of the DNA with adapters. And then I think I just mentioned that real quick. So once you have your library prepared, you have this bridge amplification where you have one strand by the end attaching or to a complementary sequence on the surface of the flow cell. So and then what happens is it bends and forms a bridge, and then the DNA polymerase comes in and then add the nucleotide to the end until you have a complete what's it called DNA, double-stranded DNA. And then once that is done, you do the sequencing by synthesis, in which you have your what's called the labeled DNTPs, and as each passes through what's it called the cluster machine, it gives a fluorescent dye. So each of the DNTPs have different colors. It could be blue, T could be red, and all that. So once you have each color, you know what DNTP it's being sequenced. And this is also different companies using. Don't want to go into detail on how each of these work, but they have the same concepts. You have your DNA fragments, then you have your DNA polymerase, you have your DNTPs, the four different types of the DNTPs, and which are could be detected in real time by the Pacific Bio, and also the Oxford Nano also uses almost the same technology in sequencing the DNA. So the advantage of the NGS compared to the traditional Sanger is also the read length is longer compared to the Sanger. Sanger can only sequence short fragments of DNA at a time, but with the advent in technology, we can actually sequence a longer strand of fragments of the DNA. And that's one of the advantages that this technology brings. And then this table is showing the number of reads based on the different NGS sequencer or platform. And then you can see the Illumina has about 375 million reads per. Let me just check this out per unit. And also the PacPower also has a huge number. You have the Solid platform also has a huge read per unit. And I think I've actually mentioned this. And once you have your sequence read, and the next thing is to do some bioinformatic analysis, where you can do an alignment or you can do an assembly. So alignment is usually there's a reference with which you align it with, and the assembly is usually done without reference. So for the alignment, there are some alignment tools that can be used. We have the different tools. We have the spaced seeds, and we have the Burroughs Wheeler. And each of these tools also has sub platforms within them that you can also use in reading the alignment or in aligning the newly sequenced DNA. So we have this under the spaced seeds. We have the ELAND and then the MAQ. And then the Burroughs Wheeler, you have the bow tie, you have the SOAP 2, and then you have the WA. And then the genome assembly is also very, very important. Remember, these doesn't require a reference. to actually do that. So genome assembly is done by um, aligning overlapping sequences and then using that overlapping sequences to detect the complementary sequence. And then you do that until you have a full length of your DNA sequence that you are interested in. So it's just all about putting the fragments back together um, to get your a long read of your DNA sequence. And then these are the two assembly what's it called, approaches. We have the Brownian graph, and then you have the overlap graph. So the overlap graph allows you to have a long uses a long read assemblers, while the De Bruin graphs uses what's called a short read assemblers. As you can see in this, so there are like codons that are used to actually align, what's it called, or to assemble the fragments of the DNA together. All right, so in what field can we apply the NGS? So this technology, like I think mentioned it briefly, we have to, for human diseases, to detect if there's a genetic component to the human diseases by finding what's it called, variant. Can also be important in drug design in the pharmaceutical companies. Could also be useful in agriculture. Be useful in evolution if you're interested in that. So here we'll be looking at gene expression analysis. We're looking at what genes are being expressed in certain conditions. So so these are the different ways to apply next generation sequencing. And then the next generation sequencing applications, you can do what's it called a whole genome sequencing, which they did for the patient, like took them about 5 hours to complete that. You can do what's it called, a targeted resequencing, and you can also do a RNA sequencing. So the whole genome sequencing has to deal with, I could say, the whole genome. So the entirety of the genetic code of an organism. So it could be for a particular patient, it could be for zebrafish, and also they did for here they did for zebrafish, and also they've done for human. And most recently, they were able to come up with a gapless human genome, even though it's not 100%, but I think it's very close to 100%. So this is longer and is the most comprehensive method for analyzing the genome. And we know why this is very important. So this is very instrumental in identifying disorders that are inheritable, and also to characterize driver mutations, especially in complex diseases like cancer and other mental disorders. And the good thing about the whole genome sequencing is one of the advantages is it provides a high solution base by base view of the genome. So you have basically you have everything been sequenced. It captures both large and small variants that might otherwise be missed. It also helps to identify variants that could also be important for what's it called, follow-on studies of gene expression and regulation mechanisms, and also can help to deliver large volumes of data in a short amount of time to support assembly of novel genomes. In the case of the targeted resequencing, just by the name, so you're targeting a particular region of the genome, not the whole thing. So this is very useful, and once you know the region you're actually targeting, because you can do that instead of doing the whole genome sequencing, you can just do a particular genome, and of course you get your result faster. You can easily identify variants within that particular region instead of going through the whole genome. And again, the last thing it produces a smaller, more manageable data set for faster, easy analysis compared to the whole genome approaches. And one other one here is when you're doing the exome sequencing, where you just look at the protein coded region of the human genome. And this is about 2% of the human genome. So instead of going 100%, you can just look at the 2% part of the genome and And then we all know that the coding part contains more than 85% of known disease relevant variants. So this could be useful if time is not on your side when you're doing your research. So we have the DNA sequence, we have the RNA sequence, which, I mean, we don't have to spell the order of the DNA, RNA, and then you have the protein. So you can do the DNA, you can do the RNA and your transcript. So in the case of the what's it called, RNA sequencing, we're interested in the mRNA, right? So you will get the from the exon, you only get the exon out, and then you do the short range, which are the fragments, and then those are assembled into a longer stretch of your what's it called, mRNA. And this is how the NGS is applied for gene expression analysis. So we know how you have your DNA, which is then transcribed into your RNA. And then the RNA goes through what's it called, processing, where it has the polyadenylation, and then splicing process where you remove the introns. So only the exon get what's it called, transcribed. So you have your mature RNA, which before leaving what's it called, the nucleus or the cytoplasm of the nucleus, before it's been translated into the protein. So you have your mature mRNA, which contains just the exonic regions and all that. So you have this region, which is then translated into a protein, which is an amino acid sequence. So this mature RNA to protein, which is at this level where you can actually detect the expression of genes. It's very useful in detecting variants as well. So the RNAseq, which is the RNA sequencing, like I said, uses the NGS technologies to sequence the transcriptome and conduct gene expression analysis. And long RNAs are first converted into a library of cDNA fragments, which in this case, you have to convert the long RNAs into coding DNA fragments. And then you repeat the process of how to sequence, where adapters are then ligated into the ends of those fragments. And then you do, what's it called? And then you do the what's it called, the polymerization, where you get your before you get your long what's it called, reads or the long DNA sequence. So the resulting sequence reads are aligned with the reference genome or a transcriptome. And this can help to generate a base resolution expression profile for each gene. So this is this is an example. So you have a nucleotide position, and then you have the what's it called, RNA expression level, and you can see at this position, you can see that this particular residue or base is kind of expressed compared to some other lower levels. So this could be for what's it called, to have a base resolution expression profile. And in more clinical settings, so one way to actually look at that is if you have two samples of interest, you have condition 1 and condition 2. So condition 1 is normal colon, and condition 2 is a tumor of the colon. So what you do basically is to isolate the RNAs from these two samples, and then you have you ligate them, and then you do the polymerization, and then you put them on the surface of a flow cell, and then you do the reading. So basically, by the time you're done with the reading, what you're interested in is to see what genes are expressed in condition 2 as opposed to condition 1, or what genes are expressed in condition 1 as opposed to condition 2. So once, let's say for example, of course, both cells will have gene A. Let's say for example, there's gene A present in both cells. So you have to look at the expression level of gene A in condition 2 against the expression level of gene A in condition 1. If there's a differential in the expression of this gene, and then you could tell if, for example, it's more expressed in condition 2, then you could tell that overexpression of gene A is the driving cause of colon tumor of the colon. All right, so this is why we sequence RNA for functional studies. And I think I already looked at an example in the previous cell. You're looking at the treatment of drug against a cell line where the drug is not applied, just to see how effect, what's it called, a drug is. If you look at how some genes are expressed in response to the drug being given to the cell, as opposed to how they're expressed when there is no drug given. And you can also want to look at the wild type versus a knockout mice. You can also predict transcript sequence from genome sequence. Um, And also, most times, some molecular features can only be observed at the RNA level, which is why it's important to actually sequence RNA because of the different alternative splicing, which could lead to different isoforms. So this is why it's important to sequence RNA. And another reason why it's important to sequence the RNA is to also interpret mutations that do not have an obvious effect on the protein sequence. So you could be looking at some regulatory mutations that might affect a particular mRNA isoform, and you can look at how much this effect is also taking place. And also, this allows you to prioritize protein coding somatic mutations, which are often heterozygous. So for example, if the gene is not expressed, a mutation in that gene will be less interesting, as opposed to if the gene is expressed, but only from the wild type allele, this might suggest a loss of function. And lastly, if the mutant allele itself is expressed, this might suggest a candidate drug target. So these are reasons why we sequence RNA. So this is just comparing the different applications and different technologies, microarray and RNA-seq. For the RNA-seq, you don't really need a lot of RNA input to mount. Resolution is high. The background noise is low. The cost of RNA-seq is also low. And it can be applied to different isoform, can be applied to different allelic expression, to some rare and new transcripts, some non-coding RNA, and to help to discover the range of a gene. So the RNA sequencing has more advantages than using the microarray. So one of the challenges is the sample that you're using. You have to look at the quality of the sample, you need to look at the quantity of the sample, and also how pure are the sample. So you want to reduce as many noises as possible to have a very pure sample. And also you have to make sure that the sample that has been collected is of high quality and all. And then the relative abundance of RNAs do vary. They really vary widely. Like you have orders of magnitude difference of 10 to the power of 7. And that it's one of the challenges when it comes to RNA sequencing. And lastly, RNA, as you all know, is very fragile because it's highly unstable compared to what's it called, to DNA. That is because it's easily degraded by a protein. So this is the workflow for RNA sequencing. Um, you have your RNA or data acquisition. You do the mapping, which whatever methods you want to use to assemble. Remember, we have the long read method and the short read method. So you do the summarization by coded sequence, by exon, by the gene span, or by the junction reads. You perform a normalization now across the sample. So you want to do that to make up for outliers in the data set. So normalization is very important. And then you do your differential expression testing, which kind of lists genes that are differentially expressed in, for example, a diseased cell sample compared to the control. And then, of course, lastly, you do the interpretation and then you validate the outcome. Okay, so did mention this already. This is just the same way you do your sequencing, you do your read alignment, you identify genes that are differentially expressed, and then your visualization. So using the different methods, like Dr. Teng said, a pipeline in R known as the bigPint is what I'll be introducing today that can be used to visualize DEGs, which are the differentially expressed genes. Okay, so just gonna, because so time. All right, so let me talk about this one. So this is IGV browser. This is also used to visualize RNA-seq alignment. Um, so you have, this is an example where you have single reads that are spliced, and you can get the coverage scale. You can look at the coverage. There's so many things that you can look at the position of what's it called, the RNA. You have the positive strand versus the negative strand, and then you can also look at the gene track at the bottom. So this it's free, freely available. It's an open source, the IGV browser, which you can use to visualize RNA-seq alignment. So this is another interesting part of RNA sequencing, where you want to know the reads per kilobase of the transcript per million mapped reads, which is the RPKM. These are just some interesting terms. Interesting terms using RNA-seq. So basically, the ratio of these two, for example, the ratio FPKM to the RPKM, it's basically used to normalize for the gene size and the depth of the library. And so we'll talk about normalization when we get started um, with the scripts on R. So also good to know that when you using the when you're doing for performing the RNA-seq, the relative expression of transcript is also proportional to the number of the cDNA fragments that originated from it. However, the number of fragments is also biased towards larger genes, because higher genes could have relatively higher expression than normal, which it's not good for when it comes to looking at DEGs when it comes to larger genes. And the total number of fragments is also related to the total library depth as well. So, but in an attempt to normalize the gene size and library depth, so we perform this ratio, the FPKM to the RPKM ratio. Okay, so let me just talk about the data visualization of RNA-seq. So there's so many plots that can be used. We have the box plots, we have the scatter plots, we have the comma bound plots, which looks at some of some quantitative features of the DEGs. So you have volcano plots as well. You have the bar plots, or in the case that I'll be we'll be visualizing, we'll be using some box plots for our data sets this afternoon. So there are so many plots that can be generated when it comes to what's it called? Different differentially expressed genes. You can also use heat maps, clustering. We will talk about that. And then I will show you all that, and we can also do it together. And these plots are made based on two conditions, C1 and C2. And a very good example of what's it called, the cluster plot, which is is this helps in the hierarchical clustering of the DEGs. And this makes it easy to interpret DEGs. For example, if you look at the red DEGs, seem to be downregulated, while the blue DEGs are upregulated in the D7 group. So the C are the control, and the D7 are the experimental. So, so within the control, you have 3 replicates, and within the experimental, you also have 3 replicates. And each line, starting from C1.1, represents a gene. All right, so from this sample, there's about 100,000 to 200,000 genes, but out of all that, these are the few genes that were differentially expressed. 172 genes, as you can see, are downregulated from the control to the treatment group, while cluster 2 genes, which are these blue line genes, kind of upregulated. As you can see, the line, like I said, each line represents a gene. You can just trace one line. You see that the count increases as you move towards the right. So these are some of the volcano plots using the bigPint, what's it called, the bigPint program. So these are what's it called, quantitative features of the genes. So once you have your differentially expressed genes, you can calculate the p-values. The can do a log 10 of the p-value. You can also do the the FDR as well. So there are 5 data metrics used to quantify or quantitate the differentially expressed genes. And once you have your data metrics, which contains those values to quantitate DEGs, you can plot your volcano plots. As you can see, also the red and the blue. Remember, the red are the downregulated and the blue are the upregulated. So they're on different on side. You can see the red, they kind of skew or away from the rest of the genes. So these are different plots or visualization that you can use to kind of actually, if you're looking to publish some of your findings, express your DEGs and all of that. So all right, so here, like I said, we'll be using R specifically to visualize some of our DEGs. I'm not sure if you've been given what's it called, the file for, sorry, the file for, that contains the different the genes that we're looking at, and then we'll work on that, and then together we'll create some really interesting visuals for our DEGs using the R package. So we'll be using a bioconductor package. I'm going to walk you through how it's installed. I'm sure by now you already know how to install packages and also library them, and then what's called again? Yes, and then that's it. So if you have any questions, you can raise your hand or you can just speak through the microphone. Let's see.